Hello friends, welcome to BMH Learning. In this video, we're going to discuss about self-splicing introns. In most cases, the RNA is cut and spliced together by a spliceosome, but in a few cases, the RNA can perform the splicing reaction on its own, and this is basically achieved by introns. That's why self-splicing or intron-mediated splicing. Group 1 and Group 2 introns are two distinct classes of self-splicing RNA molecules that contain their own active site for intron removal and exon ligation. These two classes of ribozymes are distinguished by their mechanisms of splicing and by their unique structures. Both classes of self-splicing introns perform two consecutive trans-esterification reactions in the process of exon ligation. Group 1 intron mechanism. The first step of splicing in a Group 1 intron involves nucleophilic attack at the 5' splice site by the 3' hydroxyl of an exogenous bound guanosine cofactor. This reaction adds the guanosine onto the 5' end of the intron and releases the 5' exon. In the second step, the 5' exon attacks the 3' exon boundary. This releases the intron and ligates the 5' and 3' exons. The result of these reactions is that the flanking exons are ligated and the intron is released as a linear molecule with an uncoded G at the 5' end. Group 2 intron mechanism. A Group 2 intron utilizes an internal nucleophile for the first step of splicing. The 2' hydroxyl of a highly conserved bulged A nucleotide of the intron attacks the 5' splice site. This results in the release of the 5' exon and formation of a lariat structure whose 5' end of the intron is covalently attached to the 2' hydroxyl of the bulged A. The second step of splicing involves attack by the 5' exon on the 3' splice site. This results in ligation of the flanking exons and release of the lariat or linear intron. This was all. Thanks for watching.